So we'll just go on and wrap up. The last topic that we'll discuss today is really more of a lead-in to tomorrow. So the whole reason you guys are here isn't to write the next great TMZ spouse extractor, though we'll see how well we do tomorrow. But you have interesting biomedical applications that you'd actually like to tackle. So you've been introduced to the sort of mechanism, how to use Snorkel. Now it's to start thinking a little more abstractly about how you formulate a task that is suitable for Snorkel. So we'll just give this a preview today because it'll form most of what we'll talk about tomorrow. And I've distributed, so hopefully everyone's grabbed this. If not, maybe we can hand some out. But there's a case studies handout. Who doesn't have this? So I just printed out, we'll send, there's a link in here, so don't worry that it doesn't work. It's just to a Google Slides thing. But it describes this format for a case study. So what we'll do, I'll broadly describe a case study today, and then tomorrow we'll actually walk through a case study where we're given a problem, and then we have to decide, like, ooh, how would I design a Snorkel schema? What would be the entities and relations I would use? How would I use these facts at the end of the model? So we'll do that in real depth tomorrow. But just to give you guys a sort of flavor of what we're thinking about so you can think about your project tonight and be prepared for tomorrow, we have these sheets, and I'm going to walk through the broad points just in a lecture. And at this point, we're late in the day. We've been beating our heads against the wall to write labeling functions. So please, this section is more interactive perhaps than some of our other sections. So please, if you have any questions or ideas, just go ahead and throw them out, and we can try to make this a little more interactive until we get to the welcome reception. Alex asked if the task of looking at an application and formulating a schema of entities and relations to extract for some information task locks you into Snorkel in the sense that it's Snorkel specific, and it's absolutely certainly not. This is just a broad way of thinking about information extraction. You can apply it to really any system. It's just a way of thinking about, in our context, machine reading, but broadly any information extraction task. So that's a good point. Though we hope you use Snorkel, since you've, it's a super powerful tool. Hopefully you're convinced of that today. Yeah, you might have heard of it. Yeah. All right, great. So this is just me sort of like, when I think of writing a new Snorkel application or any information extraction application, the two questions I always ask myself are, what am I actually extracting? And like, once I have it, like what utility does this provide to me? And this gets to this dark data tip of the iceberg question or perspective that Stephen presented at the beginning of the day, is that, is this information that I'm going to spend some amount of effort? Snorkel makes it easier, but it's still some amount of effort. Is it worth extracting? What value does it offer me at the end? And you should really think very carefully about these questions rather than just diving in willy-nilly, because it is an investment in time to build this pipeline. And it's better, your schemas and the things you formulate to extract will be much stronger if they're very concretely linked to an objective at the end. We just find this is just a much more solid perspective to do. And I'll walk through a couple concrete examples as we go through the day. So the sheet I've passed out, I just give you, I just repeated on the first page those 2 points I said. And what you'll be given is essentially a blank template that will ask you to fill out. And you'll fill this out in detail tomorrow. But the basic structure of building an application, I've split it out into 6 points. One is sort of providing a motivation, like why is this application a big deal? Why is it worth doing? A task overview is specifically, concretely what you want to do. And data set overview is a question of what am I applying all of these sort of pipeline I build, what data I'm going to analyze. Is this going to be PubMed abstracts? Is this going to be EHR records? Like, you sort of think about specifically the data that will go into your information extraction system. And then I've bolded the fourth item, which is the schema design, which I would argue is one of the most important things to think very, very carefully about. And it's often somewhat hard depending on what problem you're approaching. And at the end, validating your extraction models and then validating their utility at the end. And these are usually, validation requires collecting domain experts, like building some hand-labeled data, and motivating, the purpose of that data in some real-world application. So this is pretty high level, but I'll just start with the schema, and then I'll walk through a little bit this printed out example that I've included with a task. So schema, I use schema, probably the best framing is sort of like a database perspective where you have some formal representation of sort of atomic categories, right? So one big thing here is entities. As I described at the beginning of the day, an entity would be like conceptually could be a person. And a relation would be some connection between entities. So like the spouse relationship is a relation on top of people. And in Snorkel, that's sort of how we started the day defining candidates. That's the most elemental thing that you are building your application around. So it's worth thinking very, very, very carefully about what that schema is, because it'll be the sole thing that you write labeling of functions around. It'll be what you extract and use to populate your knowledge base. So it'll form some load-bearing component of your analysis at the end. So sometimes this isn't the most evident, readily apparent thing to do. For some of these examples like spouse or chemically induced disease, that's the CDR example, they sort of nicely break down into a relationship between two people, or a relationship between chemical and disease. Or maybe if you're looking at EHRs, you want to model a side effect relationship, which could be between a drug or a symptom and sign. And these are all sort of common tasks that people have thought a lot about for a long time to formalize into categories. But from when you guys gave your pitch this morning, a lot of you are thinking in sort of different headspaces. You're all researchers, you're doing novel things. So a lot of times, some of the categories that you're looking to extract or things you want to draw relationships around aren't necessarily, no one's like plowed ahead and made up these sort of nice categories and representations yet. That's sort of what your guys' job is to do when you're using Snorkel for your task. So I will just walk through this one page of a case study on mining gene-protein interactions, gene-protein interactions with chemicals. This is a real task, actually. And I'll just sort of explain like what goes into each of those 6 slots. And feel free to stop me and ask any questions. This is a very, very simple case study because the sort of entities and relationships are clearly mapped out. But I hope it gets you guys thinking about how we would approach tomorrow, where we'll go through 1 or 2 other case studies where the entities and relationships probably aren't as clear. And it's sort of more of a challenge to think about how you would model this problem in a way that would make Snorkel very useful and a good fit. Okay? All on the same page? Okay. So the CDR task, the chemical disease relation task, it's pretty informed by this sort of as a group or area of research on comparative toxigenomics data set. And this organization has put a lot of effort into curating data sets and knowledge bases that capture relationships between like chemicals and diseases, or genes and proteins and chemicals, and this sort of rich space of things like, we know the environment probably has a huge effect on our health. The chemicals we are exposed to from day to day could have huge implications for our health and the causes of disease. That space of discovery, of finding those relationships, is changing dramatically every year as we publish more and more stuff. So there's a real dire need to be able to have this sort of infrastructure to mine this information at scale, at the pace of published literature. So that, what I've just told you, is the motivation for wanting to do this machine reading task for something like finding associations between gene proteins and chemicals, right? So you can immediately imagine this task fits quite nicely in some of the projects that people have described here, where you want to maybe look at PubMed, PubMed Central, maybe you're looking at full text, maybe you're looking at abstracts, but maybe you want to pull out these relationships between proteins, genes, and chemicals. And that task fits in very nicely into the same thing that we've looked at today with spouse, where instead of modeling a relation of two people, it's a relation of a chemical, or a relation of a chemical name and a protein or gene name. And we could get a little finer point on it. We could say that maybe it's two relations between a protein and a chemical and a gene and a chemical, but fundamentally the entities are pretty apparent in this task, right? We've all sort of, anyone who's done any bioinformatics work kind of agrees that these are categories, and you can certainly subdivide things like proteins or genes into finer and finer categories, but at an aggregate level it holds together and makes a lot of sense. So the task overview would be to simply mine all of these relationships from something like PubMed Central. So have a machine reading system that could run fancy and at scale, and maybe every night analyze 1 million articles and sort of update a known knowledge base of these relationships. And think about the sort of utility that would be is if it really did run, and updated every week, how powerful a thing that could be for many different contexts in bioinformatics. So the data set, as I said, maybe you're looking at all open access journals. I think the actual, I listed 3.7 structured XML articles. That's just from PubMed Central Snapshot. There's a paper that came out a couple years ago that tried to estimate the entire space of open source literature, and it was something on the order of 140 million articles. So if you really, really wanted to say, I'm going to ignore all of the pay-to-play journals, I'm going to use this vast space of open access, that's 140 million articles you could potentially mine for all kinds of sort of interesting scientific insights. Item 4, the schema design. This is simply the candidate class definition that we've talked about a couple times. This problem very nicely lends itself to something clear in the form of gene, protein, chemical, right? Those are clear categorical concepts. When we talk about tomorrow, we'll see when those entities aren't so clear, how we could perhaps design a similarly meaningful schema. Yeah, so the schema is pretty easy to justify, right? So it makes sense to other people. Like if I gave you essentially this sheet and asked you to interpret this problem, it would probably make good sense to everyone in this room that why I picked genes and proteins to draw relationships between. So you could clearly see, interpret that schema, and you could interpret its value as a task. To validate it, if you were to build an information extraction pipeline, the most reasonable thing to do is have someone label a small annotation, validated annotation set. So you get a bunch of domain experts, and they build a collection of human annotated examples. And in the classic setting, you would use some of that to train a model, and then you would validate it using some subset of that. What we've learned today is that you can use all of that in some sense to validate it, right? You can use the set of human labeled data to tune your labeling functions, and then some other holdout to evaluate as a test set. And the big argument of Snorkel is that now that is an order of magnitude less data, training data you need, but it opens up this world of much more interactive, more software-like annotations, and the ability to train very sophisticated, fancy, state-of-the-art deep learning models, and really take advantage of a lot of the advances in machine learning. And then finally, for external utility, it's, I'd say, a clear case that you say we need this information in a structured knowledge base. This is the whole point of science, is to increase our understanding and have this information ready for other analysis. So imagine being able to pull an R data frame of every known association between chemicals and diseases with some sort of probability associated with it. Think about the types of science such an artifact could enable. Okay. So that's sort of my chemical disease or chemical protein gene pitch. This same template is what you should keep in mind when thinking about your application. And the part you should really, really think a lot about is the schema. Like, what are the entities? What are the relationships between those entities? And that probably will be challenging in some people's projects to really come upon a robust schema that you can use in, as Alex pointed out, a bunch of different information extraction settings. But it's worth thinking about that and how you will use that schema, because it really will make a much more tighter application in the end. I mean, just to point out, Steve had a great example from one of our collaborators who's doing work looking at stents and deep vein thrombosis. You know, there has never been a randomized clinical trial with stent sizes. There's such a huge space of potential with retrospective observational studies using EHR data that you can pick these sort of super important questions, formalize a schema where you're looking at stents and their sizes, maybe where they are on the body, and extract that from some huge collection of ER data. So like the VA, Veterans Administration, which we work with some in the Mobilize Center, they have something in the order of 2 or 3 billion patient notes going back to the 90s. Think about the space of potential retrospective clinical trials you could do across an crazy space of potential medical devices, potential drug interventions. All that stuff lives in unstructured data. That's a huge opportunity to do amazing science if we can unlock this ability to sort of pull stuff out of text. And to think concretely, I want to do a retrospective observational trial. I want to test stent sizes. We do not have that information. Plug it into Snorkel, and now I can ask these questions. Now I generate a data set that dumps right into an R data frame, and I can do all the sort of standard analyses I would as if that was ready to me in a normal database. Oh, absolutely, yeah, absolutely. So we're, strictly speaking, we're doing classification now on relations, but like I have work, Snorkel naturally lends itself to predicting on a single entity. Yep. Yeah, we have actually support for categorical predictions now. So I sort of haven't formally discussed this, but if you look in both the workshop directory and the advanced tutorials on the Snorkel GitHub page, there's a huge space of advanced features. Stuff like categorical variables where you're not doing binary classification. There's a demo notebook on that. Doing really intense grid search or hyperparameter tuning, there's a notebook on that. There's a notebook on using BRAT to develop gold annotation labels. We sort of have provided the tools for doing all of these parts of the full stack. We just haven't had time today to sort of go into every detail. But we're absolutely not limited to binary classification. Just for didactic purposes, it's easier to explain. So you might ask, should I totally buy into the Snorkel thing? There is sort of a vision for where Snorkel's going and sort of how it lives in the hierarchy of machine learning stuff. And we've been a very machine reading information extraction focus, and Snorkel's probably going to be a little more broad, broader even than that. That doesn't necessarily preclude it fitting in to an information extraction paradigm. But we'll talk a little more about it later tomorrow.
I was just going to chip in briefly that if you look at the materials online, there's a lot of stuff about like generating training data and stuff like that. So, a lot of the tooling and really all of or majority of the applications we've been working on with collaborators have to do with this information extraction problem. At the heart of it, as you've seen today, is training these models. It's training this end machine reading model. And so Snorkel can be used to train other models, but again, a lot of our tooling is based around this use case. So that's sort of how to connect some of the material you'll see online if you poke around the website with what we're solely focusing on today.
Yeah, so like a year from now, like images and speech, and there'll be just a huge space of other like phenomenal things that Snorkel will be able to work with.
And again, what you were doing today was a classification problem. It was, I have this candidate mention, is that a spouse mention or not? So Snorkel can be used for other classifiers, but this is sort of a problem we care about and that we've built a lot of tooling for. So that's why we're, focused on that. Essentially, there's no support for regression right now. There's support for categorical, but
so I could potentially say discretize it into say 100 categories or?
Yeah, sort of, except that it's a little bit of a mismatch in general. Categorical problems assume that you can have like totally separate buckets, not that they're on some range. But we can talk more offline about that.
That was just a question for our viewing audiences at home about Snorkel's ability to do regression problems versus classification problems. And as it stands now, Snorkel is framed as classification, so it's a bit of a mismatch. But there's certainly interesting questions there that we can talk about further. So.